Welcome to the Deep Dive. Today we're tackling, well, a really fundamental question in biology. How do we figure out if two species look alike because they're related, shared history, or if they just stumbled onto the same solution to a life problem independently?
Right, that's the core challenge. Is it ancestry or is it adaptation filling a similar niche? Our mission today, using Strickberger's evolution as our guide, is really to get into the evidence scientists use. How do they tell the difference?
Okay, so let's unpack that. The sources lay out three main patterns, sort of like different paths as evolution can take to create similarities.
Exactly. First up, the most direct one, homology. This means the similarity comes from a recent common ancestor. Pretty straightforward, like why you might share features with your cousins.
Okay, shared ancestry. But then it gets trickier with the other two patterns, right? Where the similarity pops up independently. We've got parallelism and convergence. Sometimes convergence is also called homoplasy.
That's correct. And the key difference between those two really lies in the starting point, the raw materials they work with. Parallelism happens when similar features evolve, but in lineages that are already related.
Related. How closely?
Well, they share a more distant common ancestor, so they often inherit similar developmental or genetic pathways, sort of like having similar blueprints to start with, even if they build slightly different houses later.
Ah, okay. And convergence.
That's when similar features evolve in lineages that are basically unrelated or only very distantly related. They face the same environmental challenge, the same selective pressure, and they arrive at a similar solution, but using completely different biological toolkits.
Right. And that's the big challenge, isn't it? Looking at two things that ended up looking alike and trying to figure out how they got there. Was it path A, B, or C?
Precisely. It's what our source calls the apples and oranges problem. You can't just assume similarity means shared ancestry. You need solid criteria.
So let's build that foundation. We start with homology. You mentioned Richard Owen back in the 1840s. The key was the underlying structure.
Exactly. For Owen, organs were homologous if they shared the same fundamental structure and position, even if their function changed dramatically. The classic example is the vertebrate forelimb.
Right, the humerus bone, that single upper arm bone.
Yep, it's there in a monkey's arm for climbing, a mole's forelimb for digging, a seal's flipper for swimming, and a bird's wing for flying. Same bone, same position relative to other bones, but wildly different jobs.
So, same underlying plan, different function. That's homology. And if the function is the same, but the underlying plan is different.
Then we're talking about analogy. Analogous structures do the same job, like flight, but they're built completely differently. Think bird wings versus insect wings.
Bird wings built on that internal skeleton we just mentioned.
And insect wings built on an exoskeleton framework. Totally different origins, different structures. That kind of similarity analogy arises through convergence.
And this idea of homology, it isn't just about big bones, is it? The sources say it applies right down to the molecular level.
Absolutely, it's hierarchical. At the gene level, we talk about orthologous genes. These are genes found in different species that are similar because those species inherited them from a common ancestor.
Okay, shared between species due to ancestry.
Then you have paralogous genes. These arise from gene duplication within a single species. So you end up with multiple copies of a gene or slightly different versions within the same organism's genome. They are homologous to each other because they came from that same original gene.
That sounds related to another type of homology mentioned, serial homology. That's about repeated parts within one individual.
Exactly, like the vertebrae in your spine. You have cervical vertebrae in your neck, thoracic in your chest, lumbar in your lower back. They're different, but they share a fundamental vertebral structure. They're serially homologous. Often this reflects underlying gene duplication and developmental patterning.
Okay, let's circle back to that tricky pair. Parallelism and convergence. Both involve independent evolution of similar traits. Why distinguish between related lineages doing it versus unrelated ones?
It matters because it tells us about the potential and the constraints of evolution. In parallelism, because the lineages are related, they often have that shared developmental toolkit inherited from a more distant ancestor. This shared toolkit can sort of channel evolution down similar paths when faced with similar pressures.
Like the marsupial wolf in Australia and the placental wolf elsewhere. Both mammals, so they had a shared mammalian starting point.
Precisely. They independently evolved wolf-like features, the skull shape, the teeth for predation, but they likely use similar ancestrally shared developmental pathways to get there. That's parallelism.
And convergence is when there's no shared toolkit for that specific trait.
Right. Think about cacti in the Americas and euphorbs in Africa. They look incredibly similar, thick, succulent stems, spines for defense, adapted to deserts. But they are from completely different plant families.
So they solve the desert problem independently, using totally different genetic and developmental routes.
Exactly. The similarity is purely driven by the environmental pressure, not by shared ancestry guiding the development of those specific features. That deep difference in how they achieve the similarity is what separates convergence from parallelism.
Okay, that distinction is clearer now. Parallelism uses shared tools inherited from a more distant past. Convergence uses different tools entirely. So how do we actually prove which pattern we're seeing? This gets us into the evidence, starting with comparative anatomy.
Yes, looking closely at structures, comparative anatomy helps us trace homologous structures across vast stretches of time. The evolution of the horse is probably one of the best documented examples.
Ah, yes, over about 60 million years, right?
Yep.
From the little Hierarchiopteryx, used to be called Eohippus, which had, what, 4 toes?
4 toes on the front feet, 3 on the back. Small animal browsing soft leaves in forests.
And we can anatomically trace the lineage right up to the modern horse, equus, large, single-toed, built for running on hard ground and eating tough grasses.
And the key anatomical insight is seeing how those side toes gradually reduced. They didn't just vanish. You can see the remnants, the splint bones in modern horses, which are clearly homologous to the full side toes of their ancestors. The parts are still recognizable.
And speaking of remnants, that brings us to vestigial features. Structures that are still there but don't really do their original job anymore.
Vestiges are fantastic evidence for ancestry. They're structures that have lost most or all of their original function, but haven't been entirely eliminated by natural selection. Think about certain whales, like the bowhead whale.
Massive animals, fully aquatic.
But deep inside their bodies, they still have tiny, rudimentary bones corresponding to the pelvis and hind limbs of their land-dwelling ancestors. There's no function for them now, but they persist as an echo of their evolutionary past.
Wow, and pythons too. They have little spurs.
Yes, small external spurs near their tail end, which are remnants of hind limbs connected internally to vestiges of the pelvic girdle. Clear evidence of their four-legged lizard ancestors. And of course, humans have them too. The appendix, wisdom teeth, the tiny muscles that used to let our ancestors move their ears.
Makes sense. Okay, so anatomy shows us persistent structures. What about studying development itself? Comparative embryology.
This looks at the process, not just the final product. Back in the 19th century, Karl von Baer made a crucial observation. He noticed that the early embryos of different vertebrates, fish, salamanders, tortoises, chickens, humans look remarkably similar.
They all start out looking kind of alike.
Very much so. They share features like pharyngeal arches, which become gills in fish, parts of the jaw and ear in us, a notochord, similar vertebrae formation. They only start to really diverge and look like their specific species later in development.
And Darwin saw this as huge evidence for common descent, right?
Yeah. Absolutely. For Darwin, this similarity in early development wasn't just a curiosity. It was strong proof that these diverse animals shared a common ancestor. Their developmental beginnings reflected that shared heritage.
Now, didn't this lead to that famous, maybe slightly infamous idea from Ernst Haeckel? The biogenetic law.
Ah, yes. Ontogeny recapitulates phylogeny. The idea that an individual's development, ontogeny, is like a fast-motion replay of the entire evolutionary history, phylogeny, of its species.
Like our embryos briefly go through a fish stage, then a reptile stage.
Exactly. And while it was influential, the modern view, informed by Evo Devo, evolutionary developmental biology, is that it's largely an oversimplification, even incorrect in its strong form.
So what's the current understanding?
Embryos don't replay ancestral adult forms. Instead, the similarities we see, especially early on, reflect deeply conserved developmental pathways and shared genes inherited from common ancestors. Once a fundamental way of building an embryo works, evolution tends to tinker with the later stages rather than overhaul the very beginning.
Okay, so early similarities show shared developmental blueprints, not a speed run of history. That leads us to the final major class of evidence, the fossil record. The actual physical remains.
The hard evidence, yes. Fossils provide direct proof of past life forms and show us succession how different forms replaced others over geological time. But understanding the fossil record required a revolution in thinking about time itself.
Right, the shift from catastrophism to uniformitarianism.
Exactly. Early ideas, like Cuvier's catastrophism, suggested Earth's features were shaped by sudden, massive events. But geologists like James Hutton and Charles Lyell championed uniformitarianism.
The idea that the same slow, gradual processes we see today, erosion, sedimentation, volcanism, shape the Earth over vast stretches of time.
Precisely, and this concept of deep time was absolutely essential. Evolution needs immense timescales to work, and uniformitarianism provided that geological framework.
And with that framework, fossils started making a lot more sense, like Archaeopteryx. Found in 1861, that was a big deal.
A huge deal. It looked like a mosaic, an intermediate form. It had feathers and a wishbone, clearly bird-like features, but it also had teeth in its beak, claws on its wings, and a long, bony tail like a reptile.
Exactly what you'd predict for something transitioning between dinosaurs, reptiles, and birds.
It was powerful confirmation. And while Archaeopteryx shows a transition between major groups, the horse phylogeny shows us the complexity of evolution within a group.
We mentioned the general trend, small browser to large grazer, but the fossil record isn't a straight line, is it?
Not at all. If you look at the actual phylogenetic tree for horses, like figure 3.14 in the source, it's incredibly bushy. There are many side branches, many different horse species coexisting, many evolutionary experiments that didn't lead to modern equus. Evolution wasn't aiming for the modern horse.
And the pace wasn't constant either.
Definitely not. Traits like body size didn't increase steadily. There were periods of rapid change, likely coinciding with environmental shifts like the spread of grasslands, and periods of relative stasis. It shows evolution isn't always gradual or linear.
Which brings us to the opposite end of the spectrum from rapid change, the phenomenon of living fossils.
Right, these are lineages that seem to have changed remarkably little over vast periods of geological time. They look very similar to ancestors found deep in the fossil record.
The coelacanth is the most famous, right?
Yeah.
Thought extinct for maybe 80 million years.
Until one was caught off the coast of South Africa in 1938, and it looked almost identical to its Cretaceous ancestors. Other examples include the ginkgo tree and horseshoe crabs, ancient lineages persisting into the modern day with minimal outward change.
So evolution includes both dramatic change and incredible stability.
Absolutely. Stasis is just as much a part of the evolutionary story as change.
Okay, let's try to bring this all together.
Yeah.
We started wanting to know how to tell if similarity is due to shared history or independent adaptation.
And we've seen that distinguishing relies on understanding those three patterns: homology, shared ancestry; parallelism, independent evolution in related groups using shared tools; and convergence, independent evolution in unrelated groups using different tools.
And we confirm these patterns by looking at the evidence. Comparative anatomy reveals persistent structures and vestiges.
Comparative embryology shows conserved developmental processes.
And the fossil record gives us the time-stamped physical proof of past life and transitions, both rapid and slow.
Putting it all together allows us to build robust phylogenies, maps of evolutionary relationships based on homologous features, confirmed across these different lines of evidence.
So for you listening, understanding these concepts lets you look at two organisms and ask the deeper question. Is their resemblance a family trait passed down through generations? Or is it a case of convergent problem-solving in the face of similar life challenges?
And that leads nicely to a final thought to ponder. We see the horse lineage undergoing fairly rapid, dramatic changes as grasslands spread. But then you have the coelacanth, chilling out, basically unchanged for maybe 380 million years in the deep ocean. What is it about the biology of living fossils or their environments that leads to such profound long-term stasis? Why don't they show the same evolutionary dynamism as related groups?
That is definitely something to think about. What keeps evolution seemingly on hold for some lineages for so long? We hope this deep dive into the evidence for evolution and tracing relationships has been informative and given you a solid framework for understanding these fundamental concepts. Thanks for joining us.